Today, we would like to share with you an important topic in the field of molecular biology, the genetic makeup of Aedes aegypti, also known as the dengue mosquito. This mosquito is not just a common insect we try to avoid. It is actually the primary carrier of serious diseases such as dengue fever, Zika virus, and chikungunya. Because of its major impact on public health, Aedes aegypti has become a key focus in genetic and molecular biology research. By understanding its DNA and genetic structure, scientists have been able to develop innovative ways to control mosquito populations and help prevent disease outbreaks. In this presentation, we will walk you through the chromosomal structure of Aedes aegypti, the number of genes it carries, and some specific genes and proteins that have been studied and are now used in biotechnology, agriculture, and even the pharmaceutical industry. Through this case study, we will explore how molecular biology can be applied to solve real-world health problems and how far we have come by studying the genome of this small but powerful insect.
Today, we will discuss the number of chromosomes and the sex determination system in the Aedes aegypti mosquito, the main vector for spreading this virus. Aedes aegypti has the diploid chromosome count of 6. This means that each cell of body there are 3 pairs of chromosomes, or 2n equals 6. The chromosome structure consists of 2 pairs of autosomes, namely chromosome 1 and 2, 1 pair of sex chromosomes, namely chromosome 3. However, unlike human and many other organisms that use the XY system, Aedes aegypti has unique way of determining sex. The sex of mosquito is not determined by the XY sex chromosome, but by the presence of the M locus or M locus, which is found on chromosome 1. If the M locus is present, the mosquito will develop into a male. If the M locus is absent, the mosquito will become a female. Chromosome structure. The Aedes aegypti mosquito has like linear chromosome, which is the same most other eukaryotic organisms. The number of chromosomes is 3 pair, make the plot number n equals 3. The chromosome size is also quite large and uniquely. It contains many heterochromatic regions. This means that most of the DNA in the chromosome is native or recessive. The most important distinguishing feature, chromosome 1 contains the M locus, a genetic region that's why important to male sex determination. Without this M locus, mosquito will not develop as male. Like other eukaryotic chromosome, this mosquito chromosome has centromere and telomeres, which are important for cell stability and division. Interestingly, many genes involved in virus house are also found on the C chromosome. Let's take a look at something interesting fact about Aedes aegypti. The mosquito known to be the main spreader of the dangerous virus. First, only female mosquito bite human and transmit the dangerous virus. Male mosquito only feeds on nectar and are not involved in the spread disease. Second, Aedes aegypti is currently used the genetic model in the study of vector-borne diseases such as dengue, Zika, and chikungunya. Third, researchers are using modern technologies such as CRISPR and gene-driven genetic modified mosquito in the hope of controlling population and preventing the spread disease. Finally, the Aedes aegypti chromosome is very unique. It has a complex structure and contains many heterogeneous regions, making it different from the chromosome of the other insects.
Next, we go to number of genes. The Aedes aegypti has approximately 50,000 to 17,000 protein coding genes. These genes are segments of DNA that give instruction to make proteins and control biological processes such as reproduction and growth. Aedes aegypti has 6 chromosomes that are arranged in 3 pairs, 2 pairs of autosomes and 1 pair of sex chromosomes. Unlike in humans, the mosquito uses this M locus system to determine sex. And last, The genome size of Aedes aegypti is approximately 1.38 billion base pairs. That's larger than the genome of a fruit fly. Now, let's take a look at the number of genes in different organisms to compare of gene counts across 4 organisms. First, we have mosquito or Aedes aegypti. This mosquito has about 50,000 genes that help it fly, find food, reproduce, and spread disease like dengue. Fruit fly has around 30,000 genes. Human have between 20 to 25 genes, and last, mouse has around 22 genes.
I will be presenting on key genes and how they are applied in the field of pharmaceuticals to improve disease control and prevention. TOL and IMT pathway genes are part of the mosquito's innate immune system, which is like the body's first defense against infections. In Aedes aegypti, these genes help detect harmful microbes and trigger the production of microbial peptides or AMPs. AMP are small proteins that attack and kill bacteria, fungi, and viruses. This helps the mosquito fight off viruses like dengue and Zika before they can spread inside its body. In pharmaceutical use, scientists study these genes to understand how viruses live and grow inside mosquitoes. If we can find ways to boost these immune responses, we might be able to stop mosquitoes from passing viruses to humans. These genes are also very important in Wolbachia technology. Wolbachia is a type of helpful bacteria that scientists put into mosquitoes. It activates the TOL and IMT pathways, making it much harder for viruses to grow inside the mosquito. This means fewer viruses get passed on to people.
In order to counter the dengue disease carried by mosquito, we discovered few methods that was used. One of them is CRISPR Cas9 gene editing and transgenic mosquito technology. CRISPR is a short form for clustered regularly interspaced short palindromic repeats. It's a precise gene editing technique that allows scientists to target and modify specific DNA sequence within a mosquito's genome. This can be used to disrupt genes essential for reproduction or to insert genes that make mosquitoes resistant to virus. Next, transgenic technology involves introducing foreign genes into an organism through a process called transgenesis. In mosquitoes, this might involve inserting genes that produce antiviral proteins or peptides, which prevent the dengue virus from replicating inside the mosquito's body. While CRISPR focuses on editing existing genes with high accuracy, transgenic approach aims to give mosquitoes entirely new traits. Both methods offer promising long-term solutions for reducing the transmission of disease by targeting mosquitoes at the genetic level. This is the case study we found through our research. This case study detailing the genetic modification of Aedes aegypti mosquitoes to combat dengue inherently touch into both CRISPR Cas9 gene editing and transgenic mosquito technology. The development of genetically modified GM strains and the release of insects carrying a dominant RIDL strategy explicitly demonstrate transgenesis, where specific genes are cloned and introduced into the mosquito's genome. Furthermore, the mention of harming endonuclease genes and the potential to use GM mosquitoes carrying a gene that transcribes RNA interference points directly to the principles of gene editing, highlighting the precise manipulation of DNA. While RIDL or homing endonuclease represent alternative methods, the core objectives of the research is altering mosquito genetics to control disease. Precisely what CRISPR Cas9 offers in a more efficient and targeted manner for disabling genes through populations or enhancing intrinsic resistance to virus. Therefore, this case study serves as a foundational example of how genetic engineering, including both gene cloning for transgenesis and advanced gene editing techniques like CRISPR Cas9, is being leveraged to address critical public health challenges.
As a conclusion, Aedes aegypti is more than just a mosquito. It's a serious health threat and an important subject in genetic research. By studying its DNA, scientists have learned a lot about its chromosome, genes, and how it spreads disease. With about 15,000 to 17,000 genes and large complex genomes, it helps researchers understand how to fight illnesses like dengue and Zika. Using tools like CRISPR and genetic modification, scientists can now change mosquito genes to reduce their numbers and stop them from spreading disease. Real-world examples show that these methods can help protect communities. Learning about the Aedes aegypti genome isn't just science, it's a big step toward making the world safer and healthier for everyone.